So this video is part 2 of muscle structure. Specifically, we're going to look at the gross and microscopic structure of skeletal muscle. So again, we have some words that we should become familiar with. The first one is endomysium, and the endomysium is the fine sheath of connective tissue composed of reticular fibers surrounding each individual muscle fiber. So that's what we can see here in red, surrounding each of the muscle fibers here in black. We then get a whole bunch of muscle fibers with their endomysium together, and we surround them in a sheath called the perimysium. This is the green layer here. This is a fibrous connective tissue that surrounds groups of muscle fibers, and we now call it a fascicle. So a fascicle is a discrete bundle of muscle cells segregated from the rest of the muscle by the perimysium. We then get a whole bunch of fascicles together and we envelop them in another sheath called the epimysium. So that's this blue line on the outside. And it is the overcoat of dense, irregular connective tissue that surrounds the whole muscle. So each muscle is supplied with one nerve for innervation. So there's a nerve ending attaching to each muscle. There is one artery, and this will supply oxygen and nutrients, and one or more veins which will remove waste and carbon dioxide. Typically, all of these enter and exit near the central part of the muscle. We need to have the innervation to control activity. And because the contracting fibers require continuous delivery of oxygen and nutrients, which is used for the contraction, and ultimately skeletal muscle gives off a lot of metabolic waste, and it is that waste and carbon dioxide that is removed via the veins. Moving down a level, so this is part of a muscle fiber. Each fiber is a long cylindrical cell with multiple nuclei just below the sarcolemma. Fibers are 10 to 100 micrometers in diameter and can vary in length, with some being quite long. The sarcoplasm or cytoplasm has numerous components, including glycosomes, which contain glycolytic enzymes necessary for glycolysis. This is where we liberate energy, particularly with heightened exercise. It also contains myoglobin, which is a unique oxygen-binding protein or pigment that stores oxygen needed for muscle activity. Muscle fibers also contain other usual organelles such as mitochondria and Golgi apparatus, the sarcoplasmic reticulum, which is the specialized form of the endoplasmic reticulum, and lastly, myofibrils. Myofibrils are densely packed rod-like or rhabdo contractile elements. These are composed of myofilaments. Myofibrils make up most of the muscle volume, and the arrangement of the myofibrils within a fibre is such that a perfectly aligned repeating series of bands, a dark A band and a light A band, is evident, giving the muscle fibre that striated appearance. So a myofibril is responsible for skeletal muscle fibre contraction. It contains sarcomeres, and a sarcomere is the smallest contractile or functional unit of the muscle fibre, and it is the region of a myofibril between two successive Z discs. So you can see that we have a Z disc here and a Z disc here, and the space between those two is called a sarcomere. These sarcomeres align along the myofibril like boxcars of a train. They contain an A band, so you can see this A band here, with half an I band at each end. This sarcomere is made up of myofilaments. Myofilaments are two types, actin and myosin. So a myofibril contains a sarcomere, which is the contractile unit, and the sarcomeres contain the myofilaments. So each part of the myofibril has a different name. So the Z disc or line is a coin-shaped sheet of proteins on the midline of the light I band that anchors the thin filaments and connects myofibrils to one another. So it's the joining point between the sarcomeres. The next bit is the A band, and this appears to be dark. It contains the thick filaments, which are our myosin, as well as an overlap with the thin filaments or our actin. We need to have that overlapping structure, otherwise we have no contractile properties. The I band is lighter. It contains thin filaments only, so it only contains actin. We have the H zone, and the H zone can only be identified when the muscle is relaxed. It is the lighter region in the midpoint or the midsection of the darker A band where the filaments do not overlap. So this would be myosin or thick filaments only. And then finally we have the M line, which is the region or the middle of the H zone, and it contains a protein that holds the thick filaments together. So let's now look at each of the myofilaments in a little bit more detail. The thick filaments, as I have said, are composed of the protein myosin. Each myosin molecule has a rod-like tail and two globular heads. The tail consists of two interwoven heavy polypeptide chains, and the head has two smaller light polypeptide chains that act as cross bridges during contraction. Other than being the attachment site for actin of the thin filaments, it also contains the binding site for ATP, and the heads also contain ATPase, which is the enzyme that splits the ATP to generate the energy for muscle contraction. So the heads are quite important because they can bind actin, ATP, and the enzyme that causes ATP to liberate that energy. The thin filaments are chiefly composed of the protein actin. Each actin molecule is a helical polymer of globular subunits called G-actin. The subunits contain the active sites to which the myosin heads will bind during contraction. Tropomyosin and troponin are regulatory subunits bound to actin. Tropomyosin is the long orange band, and this covers the active myosin binding sites and blocks attachment to prevent myosin from binding and thus preventing contraction. Troponin, or the troponin complex, the bright yellow structures, hold the tropomyosin strand in place. These molecules change shape when calcium binds. When they change shape, they move the tropomyosin out of position, exposing the binding sites on the actin. Okay, so there are a few other structures that we need to learn about. The first, the sarcolemma, which we've mentioned before, is the plasma membrane of the muscle cell. It is an excitable membrane and shares many properties with neuronal cell membranes. The sarcoplasmic reticulum, or SR, is a membrane-bound structure found within muscle cells, and it is similar to the smooth endoplasmic reticulum in other cells. The terminal cisterna are discrete regions within the muscle cell. They help to increase the storage capacity of the sarcoplasmic reticulum. We have a T-tubule or a transverse tubule, and these are extensions of the sarcolemma, or the cell membrane, that penetrate into the center of the skeletal muscle. So the sarcoplasmic reticulum is an elaborate, smooth endoplasmic reticulum that mostly runs longitudinally and surrounds each myofibril. It functions in the regulation of intracellular calcium levels. So it stores and releases calcium on demand when the muscle fiber is stimulated to contract. So it provides the final go for a contraction to occur. The paired terminal cisterna form perpendicular cross channels. They are enlarged areas of the sarcoplasmic reticulum, and they surround the transverse or the T-tubules. These discrete regions store additional calcium, thereby increasing the capacity of the sarcoplasmic reticulum to release the calcium. It will then release it when an action potential courses down the transverse tubule, eliciting a muscle contraction. Because the terminal cisterna ensure rapid calcium delivery, they are well developed in muscles that contract quickly, such as our skeletal muscle. So the last of the structures that we need to discuss are the T-tubules and the triad. So remember that T equals transverse, and these tubules have membranes that contain large concentration of ion channels, transporters, and pumps. The T-tubules permit rapid transmission of the action potential or the neural impulse into the deepest region of the muscle cell. They also play an important role in the regulation of cellular calcium concentrations. Each T-tubule is associated with that paired terminal cisterna. We refer to this as a triad, where the sarcoplasmic reticulum, the two terminal cisterna, and the T-tubules come into contact. So it's a grouping of the three membranous structures. The T-tubules and the terminal cisterna provide tightly linked signals to allow for muscle contraction. Muscle contraction is ultimately controlled by nerve-initiated electrical impulses that travel along the sarcolemma. But because the T-tubules are continuous with the sarcolemma, they conduct the electrical impulses deep into the muscle cell and to every sarcomere.